you very much. It's a great honor to be here, and also a relief that I finally made it to a CIFAR event in spite of the weather. Some of you may know the story behind another attempt to visit. What I am going to do to introduce the topic is to start with a bit of history, and also which will get you tell you a little bit about why I'm involved in all of this. So if we could go, most of you know, I'm sure all of you know about the gradients of health across socioeconomic status from the Whitehall studies of Michael Marmot and colleagues, and work by others. In fact, I became part of the MacArthur Socioeconomic Status and Health Research Network, and that was really the origins of what I have, a lot of what I have to tell you about today, and certainly my interest in this. There are, of course, many causes, contributions to these gradients, which affect both mortality and morbidity. My interest is, of course, the biological side and how that interacts with the social environment and other aspects of a person's life. Now, the MacArthur Network, which included Michael Marmot and others, Nancy Adler was our chair, produced a document which is for public consumption, led to an academic treatise, which has some wonderful articles, and was a stimulus for a video which has been widely used on the topic of disparities and health. And of course, the Atchison report, which in the UK, which grew out of an interest from the Whitehall studies, advocated that all public policies are really health policies and need to be thought of in that way. And this is something that societies are trying to emulate. Those of us who are aware of this are trying to get our governments and private enterprise to do this. But one of the elements that came out of this, certainly in my awareness, was the importance of early life. And so what I'm going to tell you in sort of a top-down fashion are things that involve how important the brain is. And this is a figure you will see many times focusing on stressors, on the genetic gene-environment interactions, and then the central notion is that the brain is the central organ because it perceives what is potentially threatening, what is stressful, produces behavioral and physiologic responses. So let's sort of take this apart. So stressors in life stressors include, of course, trauma and abuse, major life events, the environmental influences, the very common influences that we experience every day in our families, our neighborhoods, our social environments, our working environments, and they produce effects. And one way of talking about stressors is to think about something using the word, which can be very ambiguous, positive stress. So you feel exhilarated from doing something that has a satisfying outcome, like giving a talk, writing a paper that was successful. And in order to do that, you have to have a good sense of yourself, a good sense of self-esteem, ability that you're in control of what's going on. Then there's tolerable stress, where, of course, something happens, major life events, but you're well prepared because of your ability to control things, and you have good social and emotional support, good material support, and you're able to weather the storm. And then there's something which is called toxic stress, which almost speaks for itself, where one doesn't perhaps have those attributes, the unhealthy, one has unhealthy brain architecture, we'll talk about what that may mean, and poor social and emotional support. And this then may be exacerbated by early life or throughout life experience of chaos, abuse, neglect, and so forth. So we'll talk about these things. But then we get back to, we come to the physiologic responses, And this concept, which we help to introduce, allostasis is a word that means the active process of putting out mediators that lead to adaptation and survival, an idea that was put forth by Peter Sterling and Joseph Ayer for the cardiovascular system, and something that we generalized with the help of the late Elliott Steller and an earlier MacArthur network to pertain to all mediators of stress and adaptation. And the concept then is that when these systems are overused and by a lot of stress or dysregulated internally with respect to each other, and I'll tell you what that means in a minute, this leads to something we call allostatic load. Now, when we think about stress, we often think about cortisol and the fact that cortisol is produced in response to ACTH coming from the pituitary gland, and that ACTH is stimulated by releasing factors from the hypothalamus, which are in turn caused to be released by neural inputs from both physical and psychological stressors. What is not perhaps fully appreciated is that the acute actions of cortisol, and for that matter adrenaline, the other stress hormone, actually enhance functions such as immunity, actually enhances adaptive immunity, promotes certain aspects of memory, promotes energy replenishment and more efficient cardiovascular function. And of course, we're probably only too well aware, because cortisol is usually talked about in a negative sense, that it can suppress immune function. That may be useful in the case of an inflammation or an autoimmune disease, but it can impair immunity, impair memory, and chronically secreted promote bone mineral loss, muscle wasting, and contribute to the metabolic syndrome. So we talk about inverted use. Now, cortisol and adrenaline are not the only mediators related to stress and adaptation. We have both the pro and anti-inflammatory components of the so-called immune system, and the widespread nature of these inflammatory and anti-inflammatory chemicals in the body. We have not only cortisol and sympathetic activity, we also have parasympathetic activity. We have oxidative stress, a little bit of which is needed for cells to function normally. We have DHEA and androgen from the adrenals. And the arrows indicate that all of these are interacting with each other, affecting each other's production. They are affecting all systems of the body, including the brain, simultaneously. And their interactions can be described as each of them, as was the case for cortisol, as biphasic and nonlinear. And moreover, there are, of course, not only unhealthy lifestyle, we'll talk about it in a minute, poor sleep, but toxic chemicals in the environment feed into this network and, for example, elevate inflammatory tone, which in turn can lead to elevated cortisol. So this is what we're talking about. Complicated, but this is really what is going on within our bodies. And it's not all bad, it's not all good. And it's the balance that is the key. And one of these factors that involves the balance is our behavioral responses to stressors, whether they are positive and health promoting or maladaptive and health damaging. And a way to think about this is to think about this term stressed out, which I think we all experience from time to time, feeling overwhelmed, out of control, exhausted, frustrated, anxious, angry even. And what happens to us? We lose sleep at night. We may eat too many of the wrong things, too much alcohol, drinking too much alcohol, smoking if we're so inclined, and of course neglecting what we know should be regular patterns of physical activity, exercise, because we're in front of our computers doing things that we're sitting, not moving. All of these contribute to allostatic load. So psychosocial stress is, of course, a major factor contributing to this condition. And then we come to the brain, and this is the subject of our research in the laboratory, and and I want to tell you just a little bit about that. One of the things we've learned over the last several decades is that the brain, the adult brain as well as the developing brain, is a lot more plastic and malleable than we ever thought it would be, that the dendrites of nerve cells is, of course, where the cell nucleus, where the genetic information resides. The dendritics of many cells can expand or contract with experience, aided by hormones in some cases, that synapses on these dendrites, the sites of input from other nerve cells, are remodeled steadily. At least a subset of them are continually being formed and replaced. They have a life, a maturation of their own. And surprisingly, there is a limited amount of neurogenesis, the generation of new nerve cells, especially in the brain region called the hippocampus, which continues in some brain areas and in elderly people can be reactivated or enhanced by such things as regular exercise. We talk about 3 areas of the brain. The hippocampus, where we a long time ago discovered the receptors for adrenal steroids for cortisol, which is important for memory and mood regulation, but also it helps shut off a stress response. We have the amygdala located down here, which is important for anxiety, fear, and aggression. It turns on stress hormones and increases heart rate, for example, in its physiologic role. And the prefrontal cortex, which is important for all manner of self-regulatory behaviors, emotional regulation, working memory, and it also physiologically helps to shut off the stress response. So if the architecture of these brain areas is remodeled in some fashion, the balance between these brain areas will change. And if, for example, the amygdala becomes overactive, this may be a good adaptation to a dangerous situation because one is more vigilant. But if this stops, if the danger passes and the amygdala is still more active, then this is an anxiety disorder or a depressive disorder. As I said, we discovered that these receptors for cortisol in the major cell layers of the hippocampus, the receptors for stress hormones end up in the cell nucleus and regulate gene expression. One of the things that happens, we discovered when an animal is chronically stressed, is that there is a release of the neurotransmitter glutamate, which is part of normal synaptic transmission, but it happens during stress. Somebody showed that if you adrenalectomize animals, they do not show this increase in glutamate. And what happens if you chronically stress an animal is that the apical dendrites of these nerve cells in this part of the hippocampus called the CA3 become shorter and less branched because of the overactivity of both cortisol and glutamate. And we can block these with certain antidepressants, with benzodiazepines. It's an active process involving both the circulating hormone and the endogenous neurotransmitter. And it's largely reversible in a young animal with a healthy brain. So this is not brain damage. Now there's neurogenesis in this part of the hippocampus, the dentate gyrus, that continues and can be suppressed by chronic stress, the same kind of chronic stress that causes these dendrites to shrink. You have to do that a bit longer. You actually get the entire hippocampus to shrink and become smaller. Other people have shown that regular activity, physical activity, actually causes more neurogenesis and enlarges the hippocampus, and we'll come to that in a moment. Then getting back to something that goes back to the early stages of my PhD training, the idea that there are these epigenetic regulations, that histones, which are basic proteins that cause folding and or unfolding of the DNA so that it can be read or repressed, that these modifications occur as a result of acute and chronic stress. And something that we're currently studying involves how the hippocampus and other brain regions change as a result of acute and chronic stress. And what we found is that stress activates a core set of genes, but not always in the same direction, depending on the stress history. That chronic stress increases the gene response overall, and including the response to an acute novel stressor. That recovery from chronic stress alters the patterns of gene expression to that same kind of acute novel stressor. That acute stress activates a protective response to repress potentially disruptive genetic elements, but this response is diminished with repeated stress and with aging, suggesting some form of instability and potential for damage, and that individuals differ in their vulnerability to stressors. We've seen this in our animal models, and that certain gene responses appear to predict that vulnerability, and that epigenetic factors, especially those early in life but throughout life, appear to be involved. So what we're talking about then, getting back to these inverted U's, are these interactions highlighting both adrenal steroids and excitatory amino acids, but many other players, which acutely enhance activity, improve memory. Acutely, when they are at higher levels, they cause impair synaptic functioning, but this is a short-lasting phenomenon. And then longer lasting, we have what we now call adaptive plasticity, involving the remodeling of synapses, dendrites, and neurogenesis. And then with extreme trauma, as my former colleague Robert Sapolsky has very nicely shown, the same players mediate excitotoxicity from seizures, stroke, or head trauma because they are dysregulated, a bit of sort of allostatic load, or we call it overload. But in between, you have, if you lose this ability to be resilient in this adaptive plasticity, then you have a disorder that has to be intervened, treated by some external means, such as an antidepressant or other kinds of therapy. Cortisol has turned out not only to work at the genomic level, but it works in mitochondria to maintain the energy supplier of the cell to regulate its activity. It stimulates and inhibits release of neurotransmitters at the synaptic ending. It directly stimulates release of glutamate, but through the generation of endocannabinoids, which are the brain's own reason we respond to marijuana, the brain's own endocannabinoids actually inhibit the release of glutamate. So they have a kind of glucocorticoids can have a stimulatory or inhibitory function. They have checks and balances, and they regulate many different aspects of cell function over different time courses. In the human brain, we know from translational studies with brain imaging that hippocampal shrinkage occurs in major depression, in type 2 diabetes, in post-traumatic stress disorder, and in Cushing's disease, where it apparently is at least partially reversible when the hypercortisolemia is treated. But you don't have to have a disease with chronic stress over a long period of time, chronic jet lag over a shorter period of time, lack of exercise, and with a condition of chronic inflammation, there appears to be hippocampal shrinkage. Now, the positive side is that regular exercise, as I mentioned, enhances hippocampal function. Intense learning does this, and antidepressant treatments also apparently can enlarge the hippocampus. One of the most effective antidepressants is regular exercise. So let's talk briefly about other brain areas. While the hippocampus neurons shrink with repeated stress in this reversible fashion, so do neurons in the prefrontal cortex, which control self-regulatory behaviors, working memory. But neurons in the amygdala, involved in fear and anxiety, actually expand at the same time, and the animal becomes more anxious and potentially more aggressive. In the orbital frontal cortex, another part of the prefrontal cortex, there's also expansion of these neurons, which apparently makes the animal more vigilant. Now, this is all well and good, as I said, in a dangerous environment. When the danger passes, though, if this anxiety continues, then that's a problem, an anxiety disorder that has to be treated. So this is the amygdala, and one of the remarkable findings with our colleague Shona Chatterjee in Bangalore, India, is that with post-traumatic stress disorder, which is thought to involve something that happens within the amygdala and other parts of the brain, is that an elevation of cortisol, a timed elevation of cortisol at the time of trauma or right afterwards can actually prevent the development of PTSD. So that may be an example of an underactivity of cortisol, which contributes to a major problem. Sonia Lupien in Montreal has shown that in children of mothers who are chronically depressed, unfortunately, over a 10-year period, that there's enlargement of the amygdala in those children, along with elevated glucocorticoids, and this is the chronicity, unfortunately, of the depression in the mother. So here's an example, one example of the transgenerational effect of the parental maternal environment on the children. Again, illustrating the importance of early life events and a potentially very dangerous consequence of this. We also know that the prefrontal cortex is sensitive, for example, to circadian disruption and chronic stress, as is the hippocampus and amygdala, that this is reversible in young adults, including a study on human beings, on medical students, studying for their board exam, showing they had impaired prefrontal cortical function, but they recovered after a vacation. But there is loss of resilience with aging. We know that. We don't know if it can be prevented. And most importantly, and I can't unfortunately talk more about this, is that females respond differently. There are subtle differences in brain architecture which leads females, women, to use different strategies to solve the same problems as males, often doing equally well. I mean, there's very little gender differences in this respect, but there are different strategies that are used because of these differences in brain architecture. One very provocative study related to not just the prefrontal cortex is this study by Peter Generos at Pittsburgh, showing that people with low SES people have association with elevated inflammation, systemic inflammation, and also with differences in their white matter, which of course is the insulation of the brain. This is related to, more schooling seems to be protective and residing in advantaged neighborhoods. So here we begin to see the impact even at the level of the brain. This is, of course, a provocative study that needs to be studied, but it shows the potential extent to which this health income inequality and lower education and early life biological embedding get under the skin and can influence the brain. And so then finally we come down to this whole issue of toxic stress and early life abuse and neglect. And we, of course, will hear more shortly about genes, environment, about epigenetics, reactive alleles, and epigenetic modifications. Everyone is aware of the, certainly of many of the consequences of this. Chaos in the home leading to, in children, elevated levels of obesity, hypertension, systemic inflammation, risky families, lacking adequate emotional support, many of the same consequences. Lack of verbal stimulation, of course, the famous book Meaningful Differences, and how vocabulary and academic achievement diverge depending on early life stimulation. And then the famous studies of the middle class population in California called the ACE study, or the Adverse Childhood Experiences study, where the consequences with the rising ACE score included heart disease, smoking, obesity, drug abuse, high risk for AIDS, depression, anxiety disorders, anger control problems, and antisocial behavior. So we know that it has devastating lifelong influences. And then, as I said, we're going to hear more about epigenetics, which includes more than just modification of histones, methylation of cytosine residues in DNA, regulation of transcription, and these microRNAs that are very complicated and interesting. And then we come back to the hope for the future, which is really that this aspect of brain plasticity, the ability to reawaken windows of plasticity and make them work for us using perhaps even a drug or some kind of intervention like regular exercise that opens windows and then driving it in the direction we want with a top-down behavioral intervention of some kind. A lot of people to thank from my own laboratory and collaborators in the research that we do, and of course my colleagues in the MacArthur Network for Socioeconomic Status and Health, and currently the National Scientific Council for the Developing Child. Thank you very much.